So thank you so much. Can you hear me okay? I feel like I'm Korean pop singer or superstar like BTS. By the way, my initial is MTS, so I'm close enough, I guess. So thank you so much for invitation. This is my first aging conference. I want to tell you I'm not aging researcher. I just started 3 months ago some research on aging. So the conflict of interest lie, I start a company called Moonshot Bio. Why not? My last name is Moon, so I start Moonshot Bio to do something crazy, including aging in the future. By the way, Bob Langer is I believe, the most famous engineer in history, and he gave me the $100,000 in my pocket, and it's not a lot of money, but I need to use that money for the company formation. But my life completely changed because of this money, because before my wife, we married to more than 20 years, whenever I'm talking about research at home, she just said, "Oh, honey, please shut up. We are having dinner. Let's have the nice dinner, not talking about your research." Once this money came to my pocket, as you can imagine, my life completely changed. She keep asking the question. So I am synthetic biologist. I don't know anything about aging except we age. Also, I know something about, but I'm doing a lot of other projects, more than 10 projects I currently doing, funded by the mostly US government. But another thing I want to tell you is, who are the trainee, postdoc, and student? If you wanted to use synthetic biology tool in your aging research, we are looking for postdoc. And it doesn't matter, but this slide probably changed your mind, because this is my institute, J. Craig Venter Institute. Denmark is very famous with a lot of ocean and sea, but this university cannot see the ocean. This is my institute overlooking Pacific Ocean in the institute, and this is my selling point, right? So talk to me, after this presentation. So let me start with the fundamental question that is about what is life. I believe this is a very timing moment for me personally, because I asked that question for more than 30 years. I still wanted to get the answer, but actually, unfortunately, suddenly, 9 days ago, my dad actually passed away. So my entire seminar trip schedule completely messed up, because I need to go back to Korea. On around 9 days ago, I came back with 39 hours of travel to get here. And yesterday, I realized I worked too hard in the airplane to finish up, some proposal and paper. I completely off yesterday and went to Hamlet's, Shakespeare's Hamlet's castle. And then, of course, you already remember, to be or not to be is the greatest question, and that's really the right question for me right now, also for you, because you are talking about aging. And then I believe we are rushing toward death one day, and then that is about aging research, and that's why we are doing aging research. But the question I had, for 30 years, the question is a very fundamental question. So in physics, or in chemistry, or you can say the physical science, we have fundamental governing principles. I can list some of the principles over here. Energy conservation, you can deny it, right? Entropy increase, that's always true. And also I believe entropy increase, and then we are trying to against that increase by doing something that's generating ATP constantly, and then provision of ATP somehow to maintain our cell. That's against natural phenomena. And then, of course, the quantum mechanics, uncertainty principle, and chemical equilibrium, etc. So I want to answer the one specific question for more than 30 years. And the question was, what is governing principle, or what are the governing principle that is unique to biology. And then I'm travel crazily. For example, this year I counted how many days I need to travel, and I realized I need to travel more than 190 days to give a lot of seminars, including this conference. And then last 2 years, I collected the ideas and feedback from people when I meet. And then I got 7 governing principles in biology. And I summarized, of course, central dogma, evolution, biological robustness, that's the most important thing for today, topic, and degeneration, reproduction, and development, and causality. Causality is a little bit controversial, but I want to make 7 rather than 6. So I add 1. Also, the synthetic biology, the pioneer is the two people. One of them is the Jim Collins, and the other is the Michael Alloyd. And Michael Alloyd talking about causality, so I just add that point over here. So I'm talking about the biological robustness. This is a very interesting, because we wanted to maintain the homeostasis. We want to stay in that situation, because if we lose that whatever situation, we basically die. And then the biological robustness basically achieved by spending a lot of energy, of course, including the ATP. That's kind of my thought, because as I mentioned earlier, we inside decrease entropy, but outside, but entire system should increase the entropy. So that means in the cost of the increasing more entropy outside, and that's how we live. This is a very unnatural process. We living, or any system living, that's very unnatural, because we basically against physical law. And then I realized there are multiple different things we could think about to understand aging, because we are basically aging every day, and then we are going toward the death point. And then there are multiple things we need to consider. One of them is a heat generation. So I started heat generation project because not because of the aging, but I contacted by the Navy program manager, and she wanted to understand how heat generation could potentially help the diver, because diver, you probably already done multiple swimming before, but the swimming is okay, but once you get out of the water, you feel very chill, right? That feeling is not good. But that is not just kind of inconvenience, but for the Navy divers, they sometimes lose temperature, and then if the temperature becomes 35 Celsius, they start to lose consciousness, and then they they potentially lose their life. So this is a question I wanted to ask, but heat generation is also important because, the last moment, my dad actually passed away. I just touched the corpse, and then very cold. The feeling was, I still remember the feeling, and because the living system does not produce any heat anymore at the time. So there are multiple mechanisms already we understand in mammalian cell or the animal, but in bacterial system, believe it or not, if you type heat generation in bacterial system, last several decades, you may only find around handful of paper, less than 10 paper talking about heat generation in bacterial cell. By the way, I'm the bacterial person, synthetic biologist. I studied mammalian cell research a few years ago, but I still focus on the bacteria for today's talk. So we didn't understand why bacteria generate, and then this is very important because as I mentioned earlier, I want to help divers to maintain their heat or the temperature, and one way we can do is, the idea is basically something like that. I want to develop sunblock or cream or whatever, the lotion, and you put that lotion that containing some bacteria that potentially generate more heat compared to the other bacteria in your skin, and then wear your swimsuit, and then that bacteria potentially generate more heat if the temperature goes down, And of course, that's also inducible because we last more than 10 years, we developed a lot of different sensors. And then we wanted to make our bacteria in your skin, that's basically synthetically made, or GMO, basically generate heat if the temperature goes down in response to temperature drop. So, but we didn't understand how they generate heat, and then we basically done knockout experiment, single gene knockout, and measure heat generation using microcalorimeter. And then because of time, I do not go into the detail, but basic idea is a knockout and then screen and measure the heat generation. And then we've done more than a few hundred different gene knockout in bacteria, and then we found very interesting result. Out of the several hundred genes we knocked out, we only found 3 different genes, and interestingly, all of them is kinase. And as I mentioned earlier, kinase use ATP, and then we measure ATP level and all kind of other things. We now realize any cell does not want to have lower or higher ATP level. They want it to maintain ATP constant. And then if you knock out this kinase, you have the accumulated ATP, and then to make that ATP go down at whatever the average level, they basically consume ATP. Basically that's an inefficient process. In that way, generate more heat. And then I'm thinking about this one, potentially related to of course, the aging process, because we suddenly die, and then we wanted to predict when we will die, and then using that heat, phenomena or ATP, phenomena or the maintenance of the ATP or heat maintenance, we could, make some mathematical model that potentially predict the tipping point, that is sudden drop of the whatever physiological system, and then potentially also predict when we potentially die. I mean, this is kind of crazy idea, but I want to bring up. So last roughly 7 more minutes, I want to talk about some other tool you may use. As I kind of introduced to you, I am microbiome or synthetic biology researcher. I do believe microbiome do make impact on our process or physiology, everything. We do study on mental health, and then effect of the microbiota in your gut, and how that impact the brain function. And then there are multiple papers coming, talking about that one for several years. And then we actually developed lots of sensors related to that. I'll probably talking about that in a minute. But one thing I want to mention was genomics advances last several decades by using knockout tool, as I mentioned earlier. Because you knock out one gene, and then you now know the function of that gene in a single cell level. But do we have that type of tool? Let's say you have the billion different bacteria in your gut, and then let's say you wanted to knock out or kill only one guy in your gut, and then what changed? You increase your lifespan, or you live longer or shorter? If you just knock out one bacterium, you're probably going to ask that question. But we didn't have the tool to do that. We recently published one paper in PNAS. The tool is basically we use machine learning algorithm, and then use CRISPR system, and then this machine learning algorithm basically spit out the guide RNA you need to use to target only one bacterium out of, in theory, billion different bacteria in your gut. And then we demonstrate using fecal matter and then spike our strain of interest, and then we demonstrate we can kill only one guy we want to kill without using antibiotics. Because antibiotics kind of cause a lot of other problems, as I will mention later on. And then at the same time, we can do that using integration of the specific guide RNA assisted template DNA into the genome of the bacterium. We can now isolate using antibiotic selection only one bacterium that potentially beneficial for your health increase or potentially make you live longer. So you can just check out, but I cannot go detail because of time limit. But I'm talking about making some GMO, right? And then, as I mentioned, we do some manipulation here, and then that becomes GMO or whatever. And then we wanted to have, actually we also developed some biocontainment system called the kill switch. Once you've done something like manipulating some probiotic and then engineer and make that GMO, and then let's say GMO kind of make you live longer, but at the end of the day, this GMO probiotic strain will go out with your fecal matter, and then that there are a lot of people, especially European people, hate GMO, and then we developed kill switch in response to temperature change from the 37 degree to whatever temperature outside. We demonstrate bacteria completely die. But at the same time, we need to understand the problem of this bacteria. GMO go out and then in the environment, but at the same time, we need to understand we basically release a lot of DNA that is modified, including the antibiotic resistant gene in the system. If you don't, by the way, I want to ask, how many of you done cloning using plasmid that contain antibiotic resistant gene in your life just once, at least once? A lot of people. How many of you kill your bacteria after you done cloning? You should kill the bacteria, otherwise you are in jail because government will. How many of you destroy DNA after you done experiment? You done destroying? How you destroy the DNA? How you done destroying DNA? What you? Pardon? Autoclave. Autoclave does not destroy the DNA. Autoclave only denature DNA, and then once the temperature go down, this is the real air. So the only bleach doesn't. Yeah, same thing. Denature and coming back. The only way you can destroy DNA is burning entire waste. But my question to everybody, because I generate a lot of GMO, because we are the cloning lab, we are generating a lot of the bacterial strain with the plasmid in it. Last 50 years, we dump this DNA into the environment. Nobody care about this DNA. And then you're talking about antibiotic resistant spread, you blame farmers because farmers use a lot of antibiotics every single day. Second, people we blame is the clinic people, the doctors. But we need to think about this problem because we release this DNA into the environment and then this one potentially transfer into the other pathogen, giving this pathogen become superbug. So, but to solve this problem, I wanted to understand and then basically do research and then basically a lot of research, ecological distribution of the synthetic DNA, but I couldn't find any literature. So instead of kind of doing that research, I actually generate new protocol that is an antibiotic resistant gene-free cloning method. I don't have time to explain the detail, but the only thing you need to do is ask me or just go to the ARDD. That's basically a repository. You can buy some plasmid and strain with a very small fee, and then you can do this cloning without using antibiotic, chemical. In that way, professor save money, and then you don't feel guilty when you do cloning. So I can skip that on. We developed a lot of sensor, and then I just recently, this paper just accepted, and this is basically some metabolite sensor, including some neurochemical. And as I mentioned earlier, microbiota also sense, also metabolize neurochemical that potentially affect your brain function. And as you can imagine, you think brain produce neurochemical, but in fact, one of the most important neurochemical called serotonin. That's important because that makes you happy. Serotonin produced more than 90% from the gut cell rather than here. And then whenever I travel and then do seminar, I get free dinner. I feel better because serotonin somehow produced just right amount. So we developed a lot of sensor, but the one thing I want to tell you before I'm ending, I'm the not aging researcher, but you tell me what you need in terms of tools, in terms of sensor, we will deliver. That's one thing I want to promise. So because of time, I skip all of that. And the one thing I want to tell you, this is more for the young researchers. I started 3 years ago synthetic biology young speaker series. That actually become hit, and then I become very famous to the synthetic biology field, including the Ed Boyden over here, actually gave the brilliant 5-minute talk for the young speaker, and then encouraging young speaker, because young speaker are giving 45 minutes to talk about their research. And then the community actually engage and support this kind of young speaker kind of supporting thing. We have now very many speakers, including Ed Boyden, talking for this seminar series, weekly seminar series, and I become YouTuber because of that. So again, if you are bored by the conference, of course, my name, and then YouTube, you'll find my YouTube video, click, heart, subscribe, right? So we have the one Nobel Prize winner in this seminar series, and then 21 National Academy members, including Ed Boyden, and then 20 editor-in-chief in synthetic biology. I didn't know we have the 20 journal in synthetic biology until I counted. 23 funding agent director, including some European agent director, and then 9 Nature and Cell journal editors, and then 165 rising star we have. And then if we have gloomy or life is too painful, just type my name in science, and then you'll feel better. I guarantee, because this one-page paper, you'll feel better, much better. I'm chief editor of the New Biotechnology. It's a European journal, more than 10 years. And then if you send me your manuscript saying your talk is amazing in the cover letter, I'm considering your journal, and then I will consider your journal in your paper in more favorably. Okay. So thank you so much. I way over, I guess, but I'm happy to answer any question. Also, all of work is because of my student and postdoc. I done nothing, as you can imagine, traveling 190 days a year. How can I do research? But they done all the research, and the only thing I done for this project is ask question, ask how they are doing. When they kind of excited, saying we finally find this data, my contribution, the only contribution is great job. And that's it.
Thank you, Moon.
Thank you.
We've got time for 1-2 questions.
Can you put one of your initial slides about the lab, like in the beginning?
Which one?
Very first slide, like
The which one?
in the beginning, like first or second, like first slide for your lab.
For my lab. Beginning. Okay.
Beginning, just this one.
Oh.
Anybody has a question? Back there in the entrance. She's not even in the room. Come at least in the room.
So if you don't have any question, just
No, that's one coming.
send me an email. I'm happy to answer.
Yeah, sorry, I just wanted to thank you for the talk. I wanted to ask if you've delved into the microbial environment in wastewater, and especially then in lakes, and how that, you were talking about remaining DNA and remaining antibiotics in water, and if you've delved into that area yet, or if you're just dealing with...
So that's a very wonderful question. I wanted to study the distribution of those synthetic DNA in the environment. I couldn't find any literature, so I wanted to do that gigantic ecological study, collaborating with all the environmental engineer. And then I realized that takes a lot of time, also money. And then instead of doing so, I just devise a new, cloning method. But I'm gonna do that in the future because that's a very important, question we need to ask. And then, U.S. government very interesting. After I talking with them, they wanted to know how many synthetic DNA out there. So that answer your question?
Yes.
Wonderful. Thank you.
Perfect. Thank you.